Greetings to all the learners. Let us learn about DNA microarray in this video session. To define DNA microarray, it is a powerful tool used to analyze gene expression, detect mutations, and study genetic variations. It allows researchers to examine thousands of genes simultaneously in a high throughput manner. The procedure involves several key steps. The first step is sample preparation, wherein we are going to extract the RNA from the source. It can be any biological sample, a cell or a tissue. And the RNA is purified to remove contaminants. And the quality control, it is a quality check to assess the integrity and concentration of RNA using spectrophotometer or gel electrophoresis. Then comes cDNA synthesis and labeling. The extracted RNA is converted into complementary DNA, which is called as cDNA, using reverse transcriptase enzyme by the process called as reverse transcription. cDNA is then labeled with fluorescent dyes, CY3 green for one sample, CY5 red for a comparative sample if it is performed in a two-color experiment. Then the step is hybridization, wherein the labeled cDNA is applied onto a DNA microarray slide. The microarray contains thousands of immobilized DNA probes, which are single-stranded DNA sequences and specific to different genes. cDNA hybridizes to complementary probes on the array slide. The slide is then incubated and the optimal hybridization is encouraged at a controlled temperature. Then comes washing and scanning, where the slides are washed to remove unbound or weakly bound cDNA, and a laser scanner detects fluorescent signals from hybridized cDNA. Fluorescent detection includes the intensity of the signal corresponds to gene expression levels. Finally, data analysis, which is done by image processing. The scanned image is analyzed using specialized software. Normalization, fluorescence intensities are adjusted to account for experimental variations. Gene expression profiling, which includes the data is interpreted to determine which genes are upregulated, downregulated, or unchanged. This is the pictorial representation of the DNA microarray, where the normal cell and the cancer cell are compared using this DNA microarray technology. And from these biological samples, the mRNA is isolated and it is purified, and then it is allowed for the formation or for the synthesis of cDNA by the process called as reverse transcription, and then labeling. This is two-color experiment. So one of the DNA is labeled with CY3 green color and CY5 red color, and followed by hybridization, and then they are studied through the scanning, where you can see the genes are expressed either only in the normal cell or in the disease category, or the genes can be expressed in both the cells. There are different types of DNA microarrays, which include cDNA microarrays, which are pivotal for measuring gene expression levels across various samples. The next one is SNP arrays, that is single nucleotide polymorphism, aiding in genetic research. Gene expression arrays, which are designed to quantify mRNA levels essential for understanding gene activity. And finally, the comparative genomic hybridization CGH arrays, which are used for detecting genomic gains or losses important for cancer research. And applications of DNA microarrays, it is used in gene expression analysis, genotyping, drug response studies, and pathogen detection in infections. Thank you all.